Hi, thank you for joining us today. I want to welcome you to our Genetic Technology in Practice session as part of the GAGC's 2020 online conference. This session represents the first of what we hope will be a very successful session focused on academic and industry collaborations and genetic practice in an industry setting. Today we have 5 fantastic speakers joining us to talk about the work that they do at their jobs. Some of them are agriculture in nature, some of them are biotech for drug development, but all the speakers are going to discuss the work being done on their day-to-day to not only further drug development, but also to apply basic research practices into their work. Before we begin, I want to go over a couple of basic housekeeping items. So sorry. I wanted to let the session attendees know that they can ask question and answer using the chat box in their Zoom feature. Each presenter will have 15 minutes to present their slides, followed by 4 to 5 minutes of Q&A. We are going to be following the GSA Code of Conduct for this session, and the session will be monitored accordingly. This session is also going to be recorded, and these sessions will be available for viewing for 30 days after today's conference. Our first speaker today is Dr. Kenneth Bruno from Zymergen, who will be presenting his talk entitled Fungal Genetics and Automated Strain Engineering at Zymergen.
So am I unmuted now?
You are.
Okay. Thank you for letting me talk about Zymergen here today. I was very much looking forward to participating in the Allied Genetics Conference for one major reason, is that the Genetic Society of America is probably the key supporter of fungal genetics in the US. And we have a big meeting every 2 years at Asilomar, which a lot of people go to Asilomar, but in the fungal community, it's just known as Asilomar. And it's literally, it's the GSA meeting, and it's a really great community, a fairly small community of researchers, and you get to know all of them really well. And I've been attending that conference since 1997, I think. So it's been a part of my life for quite a long time. And so I'm happy to talk about filamentous fungi with some more, a wider group of geneticists, like what I'm anticipating being in this audience. So let me see if I can get a hang of switching through these things. The name of the game at Zymergen is automation. And so when I came to join Zymergen, I had experience with filamentous fungi. I've worked on fungi since the early 90s. And they were starting a company that focused a lot on genetic engineering, systems engineering, and this new kind of term, or it was new at the time, synthetic biology. And so I always, you ask the question, what do you think of when you think of those words? And you pull up these cute little stuffed animals. I mean, yeast and E. coli are the name of the game in when it comes to synthetic biology. And so there's a real serious challenge for bringing filamentous fungi into something that's close to synthetic biology, or at the very least, just automation. And so what I'm going to talk about today is how we went about adapting filamentous fungi to automation and doing strain builds and genetics. I wanted to go a little deeper into some of the specific genetics, and we will have a publication out in the next few months that'll go in more detail. But this talk is very similar to what I gave back at Asilomar. And I'm just going to go over exactly kind of how we think about automating builds in fungi. And the first thing I want to point out is that, especially with a broad audience, is that I work with filamentous fungi, and another word for that is mold. So most people, very often they think of mold when they think of filamentous fungi, and it kind of has a negative connotation. But the truth is we've used fungi for a very, very long time. So centuries ago in Asia, especially, they were making soy sauce and all kinds of different food products with filamentous fungi. And everybody's familiar with cheese, right? And cheese, it utilizes fungi there in the natural environment, right? And so we have a history of taking the microorganisms that do a job that we want, even before we understood the biology or the genetics of them, and we would adapt them to a large scale kind of industrial process, right? And you go forward to where biology gets a little more advanced, and Charles Thom, who's a really famous person in industrial microbiology, investigated citric acid production. I'm going to talk a little bit about that and kind of strains we looked at for citric acid in this talk. And also, everybody knows penicillin, right? So the penicillin story is one of the great stories about how filamentous fungi are being utilized. And the truth is, these organisms were picked and used, not because they're good in synthetic biology, not because you get really high homologous recombination like you do with yeast. They're actually can be difficult to work with genetically, but all these things were adapted because the organisms themselves could do that and not necessarily because the tools were there. So now we're in a world where synthetic biology has advanced in places where the tools were really great, and we have some great workhorses that are filamentous fungi that we need to kind of catch up. So a really interesting thing I found about the penicillin story is that when they first started making penicillin, there was a big push to make a whole lot more of it, right? So they knew that it was working, and they needed to give a lot of it to soldiers during World War II. Alexander Fleming discovered that it would be doing something in 1928. Eventually, he got some help from industrial production people that were making more of it. But even after his discovery in 28, there wasn't a successful demonstration in a patient until 1941. However, on D-Day, every soldier had some penicillin in their pocket. So what happened? How did you get from barely enough to give to a patient to really, really large amounts to give to thousands of people? Well, part of it was there was an improved strain that was found in a grocery store. And this is not an improved strain by mutagenesis. It was just a random strain that someone found and in Peoria, and they put it into the process, and they were able to make a lot more penicillin. But the truth is, the history is almost all of the improvements were done with process engineering. So they got a lot better at growing it and stir tanks and all these kind of things, and that's how they made a lot more. But then as time went on, people got to know more about genetics and more about DNA in the 40s and the 50s, and people started looking at filamentous fungi for basic genetic problems. And at some point in the story, Guido Pontecorvo suggested that you could use mutagenesis to improve the strains. And so concurrent with process engineering became strain improvement. And so we started to learn a lot of things about genes and gene functions and the power of genetics. And then there's this thing called synthetic biology. So we're back up to today's times. And well, filamentous fungi kind of got left behind a little bit in that because the tools are so much better in E. coli and in yeast. And so when Zymergen was founded, it's founded around the notion that you could utilize high automation to make large numbers of genetic edits and be able to examine lots of these and discover things that are important for your process. And so for a long time, we had this approach of radical empiricism where we would just make every strain and test every strain. And so that was my push whenever I joined Zymergen was, hey, can you do this in filamentous fungi? Can you make transformations work in filamentous fungi? And I'll come back to this in a minute, but this is a photo of the entire company back in 2015. I'll skip over this one. It was just something from my past. One of the things we learned is that whenever you do strain improvement, what companies have been doing over years was random mutagenesis. And what happens with random mutagenesis is you get improved strains because that's what you're screening for, but you also have deleterious mutations, right? And so after a certain number of rounds of mutation, you've collected too many bad things and you've possibly gone down one path where you can't improve the strains much anymore. And we find that that's what's happening with a lot of industrial processes, not just fungi, but bacteria as well. And one of the things that we try to do is go clean up those strains. And it's not always something that you can do very straightforward. In an ideal world, you'd be doing sexual crosses in filamentous fungi and back crossing against its parent, so you get rid of some of the deleterious things. But not all filamentous fungi used in industry have an available cross. And also in bacteria, it's a whole nother thing. So how do we put high throughput around this problem and go in and try to fix these individual mutations and make improved strains? And just briefly, I did this as a demonstration by comparing two different strains of Aspergillus niger that make citric acid. And there's one strain that has 44 different SNPs between a higher production strain, and it has a huge increase in citric acid. And so we went about exchanging the mutations between these two strains to figure out which mutations were good and which ones were bad. And so I'll talk a little bit about that. But and I'll go through this kind of quickly. There's a lot of steps to transforming filamentous fungi. I think a lot of people in this audience, if when you see a fungus, you're not happy if it's in your cell culture or it's on your plant culture. So maybe not everybody's as familiar with it, but basically filamentous fungal transformations are done through protoplasting for the most part. And there are a few other techniques that are similar to yeast, but for the most part, we use protoplasting. And so we had to go about taking every one of these steps and automating them. And so DNA design was something that we had in-house. Zymergen is a third of the company is software engineers, and they write all our software. The protoplasting and the transformation and screening were all things I had familiarity with, but everybody, like I said a second ago, has their kind of fears about mold to some degree if you don't work specifically with them. And I was told by my boss who hired me, as one of the founders, he said, you got to do all this in automation and you cannot contaminate anything else or we will fire you. And he was very serious about that. So I'm happy to say I'm still working here, so I haven't over-contaminated too many other processes. So how do you go about breaking down these steps into individual things? And see if I can get through this briefly. One way to think about it is if you give someone instructions on how to make a peanut butter and jelly sandwich, right? It's just bread and peanut butter and jelly and you have a sandwich. And if I told you just do that and you're a robot, you would have no idea where to get the bread. You would need coordinates and positions and volumes and every little bit of information. So it's kind of like, you know, teaching a kid to make a peanut butter and jelly sandwich. There's a lot more detail you need to give to a kid than you need to give to a colleague or another adult, right? And it's even worse when it comes to robots. So if you want to teach a robot, you have to tell it exactly where everything is and how to do it every single step. And that's one of the things we did. And I did this for demonstration purposes. If you take a typical protocol that I used to write for doing transformations, and then you break it down into individual steps, this was just one draft of it. And it went from 2 pages that a normally trained scientist could read to multiple pages of individual steps. And actually, that's an underestimation of how many detailed position changes and things had to be done to do the transformations. So to help us with that, we use something called business process modeling notation, and it also does a really good job of tying things like tracking specific pieces of data to what you're doing physically in the lab. And so we bring all these things together. And one great thing about Zymergen, I'm just one fungal guy. We got some great automation people and some really good people at planning out these things, and they help me deliver this process to fungi. And so this is just an example of how you would do it in each one of the stages where you might take a reading or make note of something like, what's the freezer stock number? It's barcoded, it's in a freezer. And where did it come from? What date was it? You harvest the spores. How many spores did you get? What was the exact culture time? And one good thing about this is that it can allow you to kind of trace back and see where things went wrong. And so you get really, really good at honing the process. And one of the things that my team or the team at Zymergen has gotten very good at is making protoplasts. And we can make oodles and oodles of protoplasts for all the fungi that we work with, and it makes doing this a whole lot easier. But we got there by being really, really careful about individual steps and tracking everything that could have gone wrong and backtracking them. And so we have multiple fungi that are on board it now, and we do genetics on all of them using automation. And so it's really working. I'll put it that way. So there's a lot of challenges to doing automation in fungi, but we have a lot of it running right now. Just another thing to consider is that whenever you're building a strain, you're going to have lots of different pieces and components like primers. If you're going to make a knockout, you're going to have components of the knockout construct and things like that. And all of that has to be traced, and you have to trace it from the beginning, because if you don't, when you go to do the QC or to see if you have what you think you had, you have to know what you started with and through the entire process. And this becomes a problem, you know, if you worked in a lab, in a fungal lab before, or just a genetics lab, you know, so you're doing 2 or 3 transformations or something could go wrong. Well, if you're doing 10 plates of transformations, 960 things can go wrong. And so that can be a real challenge. And so one of the things I've learned about working at Zymergen is it's just a different way of thinking about biology. You just can't think of individual wells and things like that in experiment. You have to think in terms of plates and groups of proteins and groups of genes that you're going after. So it's been a real eye-opening experience working at Zymergen, learning just this different way to think of automation. And here's just another example of combining the business process modeling with what we do for transformations. So, and I don't know if it'll play or not, but I had a video. This is one of the first automated things. So loud. This is what transformations now will look like. Fungi. You lay things out on a deck and you put your DNA in one, you put your protoplast in another, and you tell the robot to go mix the things in the right order, and you're able to plate them. And these are the kind of throughput that we can have. And this is just one TCAN deck at Zymergen. I don't even remember how many we have. It's in the dozens. And so the capacity for individual projects is pretty high at Zymergen, and it's very scalable, I should say. Another thing that we used for that was a big advantage for being able to do high throughput with filamentous fungi was getting a good boil prep to work for fungi. So what we can do is harvest the fungi. We have different species, some of them sporulate, some of them don't, and we were able to work out ways to do a boil prep in order to get genomic DNA template in order to test it. And one of the things to thinking about automation is that there's different kind of choke points along a pipeline, right? And so one of the choke points is getting the DNA into the cells in a 96 well plate, right? You're kind of narrowed down to how many plates you have on a deck, like I just showed you. But anytime in a process that you can open up a choke point, it allows you to think differently about how you do each of the other steps. And one of the big advantages we found is that we can screen large, very, very large numbers of cells using the boil prep, and it allows us to be okay making a lot of misses and a lot of mistakes because our capacity to go find the right ones is really so high. So I mentioned at the beginning of this, yeast is a favorite and E. coli is a favorite for reasons of molecular biology being easier in those organisms. And yeast very famously does very high homologous integration. Filamentous fungi, not so much, especially some of the species that people are using in industry. And what we've been able to do is sometimes when we need to just brute force it. So we can really just push enough numbers through, and being able to screen is one of the big advantages. This is one of my favorite robot setups that they're showing here. There's a robotic arm that moves the plates between a PCR machine, between a centrifuge, between a thing that unseals the plate and a thing that seals the plate, and also into the Echo platform, which is a liquid handler that uses acoustics. So you can set up entire 384 well plate transformations. The thing here is what we call a hotel, and there'll be a stack of, let's say, 14 plates in the hotel, and you can run all of them by just setting up the stack and checking the barcodes and saying run. And it can go and put all these into the Echo and not use any tips, and you can PCR screen or NGS screen thousands and thousands of samples. And this is one of the ways that we're able to open up what we do with fungi, is because of our screening capacity is so great, and the software people that have helped us deal with these kind of things. We also do something which I found was really a lot of fun. If you're from an academic lab and never seen one of these, we have these machines called a ZAG where you can do capillary electrophoresis. We used to call them fragilizers, I guess. And you can literally run 96 wells of a gel all at once. And we have multiple of these machines. This one here, I think, has 12 drawers. And we can literally run and check that many PCs. And we also have in our own LIMS system a way to pass, fail these QCs to see if they are what we wanted or not wanted based on PCR. And so that's another way we can do screening as well with NGS. And I'm not sure how I'm doing on time, but I'll do the NGS explanation really quickly. Okay, 16 minutes. So I will skip past this, and I can answer some questions about this later, maybe. Filamentous fungi have multiple nuclei per cell, and if you have one mutant that's yellow and there's 9 nuclei in the cell that make yellow and only one that's black, you'll still turn out black. So you needed a way to check the QC to see if it's genetically there, and we were able to do that with NGS. And for that, I will say thank you. And this, remember the first picture was the entire company back in 2015. And this was just people that work on fungi about 8 months ago, and it's bigger now. So thank you for everyone. I'll turn off my mic, or should I stop?
You can leave your mic on for now. Thank you so much, Ken. That was great.
No
if we have any attendees that would like to ask Ken some questions, they're free to type their questions in the Q&A chat box. And I can relay them to him. I have one question myself, actually, Ken.
Sure.
You mentioned you do the screening process to first create multiple iterations, and then you determine the correct strain. So for your application, how is that process determined? What is the time to determination? How is that normally handled?
Yeah. There's two main ways you can think of what is a correct strain. I mean, ultimately, a correct strain is an improved strain, right? One that actually makes more. So I did not talk about the test processes we use in this talk at all. I'm a geneticist, and I make all the mutations and things, and then we hand it off to an entire another team that just does test. And they've been able to do things like get some of the tests down into 96 well format so that we can test every one of these screens. Ultimately, that's what's the correct one. The correct ones which are improved. We also have a QC process where we check to see if the mutation that we were targeting was actually made, right? And that's kind of in my ballpark. And so we use the PCR stuff in the NGS I was showing there at the end to do that. And before we deliver any kind of clients to the strain, we test them in fermenters as well. So we have fermentation capacities too. So the whole goal is to find the right edits that improve the strain.
And is there any, are there ever improvements, so to speak, where the goal is to either streamline a process or reduce the costs of a production?
Oh, yeah.
And then for that, how long do you normally know, or how long is it before you know whether you've made the appropriate mutations?
Yeah, so basically everything we do is to lower costs, right?
Okay.
One of the things that we found is that you can do with this approach by being able to do individual mutations one at a time is that you can train the changes for either increases in yield or increases in productivity. And those are two different things. So yield is, grams per gram sugar, and productivity is basically how fast you got there. And sometimes producers care more about one than the other. And sometimes producers say, don't harm yield, but increase productivity. And so being able to do individual edits and test them, it leads us to those things in a way that you could not do with random mutagenesis. So.
Gotcha. Excellent. Thank you so much. Well, if nobody else has any questions, I'll say thank you so much to Dr. Bruno. Appreciate it. And we're now going to hear from our next speaker, Dr. Eli Rodgers-Melnick from Corteva Agrisciences, who will be speaking to us about making the most of our molecules, a computational framework for unified germplasm characterization and interference.
Thank you. Let me get to my screen here. Let's see, I'm seeing black.
We were able to see your screen.
You were? Okay, I was seeing black. All right. So you can see my screen?
Yes.
Okay, great. Thank you. So I'm Eli Rodgers-Melnick. I'm from a company called Corteva Agriscience. We actually just started this past year in 2019 as a spinoff, the agricultural sciences spinoff from the merger between Dow AgriSciences and DuPont Pioneer. And so I'm going to be talking about computationally how we've organized the legacy genetics in those and dealt with some of the challenges in combining genetics from two different companies and making sense of those companies while at the same time trying to manage an international corn breeding program. So what I'm going to mention here is, first of all, the concept of haplotypes, how we value them within Corteva, how we go about trying to place haplotypes on everything to organize our genetic diversity, how reality gets complicated when you're merging two companies, and finally, how we go toward a universal framework for germplasm characterization across all of our genetics. So the genetics that we're dealing with in Corteva go back to really before the 20th century, starting with a set of founder lines when we're talking about corn that have been organized into two heterotic groups to exploit the concept of heterosis that Charles Darwin first described during the 19th century, where we're essentially trying to breed for genetics that complement they complement each other and essentially produce the highest yield. And by doing so, we've essentially been able to increase yield in North American corn by an order of magnitude over the last century. And so from a genetic perspective, what we're talking about is starting with a set of founder diversity lines. So here what we're doing is representing each genomic segment, a haplotype by a unique color. And so that genetic diversity has been rearranged through recombination during the breeding cycles to produce the diversity that now complements each other. And so you can see that there is a rather different set of haplotypes when you're looking at one heterotic group versus the other. But when these two come together within an F1 hybrid, they produce the yield that we see in modern day field corn. So in order to make sense of these, we make use of whole genome sequencing as an initial foray. And what we'd like to do is be able to make those colored charts. And so the way that we organize these into haplotypes is, if say we have an inbred and say we've got A through H inbreds here, and we compare it to some reference, if we color the SNP blue if it matches the reference and color it orange if it doesn't match the reference, you can see how the diversity lines up here. So we have large regions where there appear to be haplotypes that match between the each inbred and the reference. And because we have a closed breeding program, there are a lot of these. But at the same time, we have regions that go back further. There's been a lot of recombination since then. And so you get these microhaplotype matches, along with rare double recombinants, which we're especially interested in when it comes down to narrowing down where genetic signal comes from in our program. And there are regions of high diversity that essentially exploit the vast amount of diversity in corn, and other regions where there is low diversity. And so if we extend this to now those color charts looking across multiple lines, what we can do is develop a hierarchy where, say, we color the haplotypes on the first inbred blue, and anything that matches that in a second inbred, we can color it the same color and give that second inbred its own color, here red, and we just continue down the line. So we can view our diversity in terms of this hierarchy, which lets us say how different family structures are impacting as, say, new inbreds become more and more popular at certain stages of the breeding cycle. And so what we end up with is these haplotypes that are defined by their sequence identity that have been defined with millions of markers. And this lets us go from sort of the initial assumption that we had in a population genetic standpoint, where we might view each founder as a unique element, but by fully characterizing these segments using whole genome sequencing, we can see that in fact the founders have some common ancestry and they share these genetic segments. And then we can go and characterize each segment in our breeding program, and the effect of each chromosome region can be predicted in new untested lines that have the same haplotype. And ultimately, if we want to make a prediction in something that we haven't phenotyped, we can sum the effects across these haplotypes to provide predictions for a trait we're interested in, such as yield. And one nice thing about working for an agricultural system is we have this closed breeding program. And so it can be relatively straightforward to extrapolate from the initial set of diversity to tens to hundreds of thousands of lines. But you know, even though we're a large company and have resources, usually we don't want to spend whole genome sequencing dollars on tens to hundreds of thousands of lines. Especially when we have, you know, multiple crops that each have tens to hundreds of thousands of instances. So what we'd like to be able to do is go from a lower density panel that we can cheaply ascertain and take advantage of population genetic features, mainly kinship among families, and then looking at narrow regions, linkage disequilibrium in small regions that go back to ancestral populations, and be able to use that to impute the information that we gain from high density sequenced inbreds to our entire population. So when I joined the company, which was before Corteva was joined, it was actually just when this merger was announced that ended up leading to Corteva. And so I was immediately presented with the complications of reality. So we have evolving marker platforms. As technology comes along, the marker platforms change, but we still want to make sense of all the legacy germplasm. We also have acquisitions that mean that new germplasm is coming in that may have different family structures to our existing germplasm. And we had this, in the case of Corteva, 2 different germplasm pools coming together. But just for reference, the lawyers didn't want me to get into specifics about where any particular company, where any particular germplasm came from. So I'll just designate them population A and population B, referring to these 2 arbitrary symbols. that made up the germplasm of Corteva Agriscience. So when we're trying to make sense of this entire germplasm pool, one thing we can take advantage of is that the different companies actually do have some common ancestral history. So the first reference genotype in corn was actually this B73, which has essentially been used as a line across the industry. This is actually a receipt that we found for when Pioneer, the progenitor company, actually purchased B73 back in 1972. So when we're talking about these two different companies, we have some common ancestry that ultimately independently went down to form our two populations. At the same time, during the course of each company's breeding cycle, there were introgressions of new genetic material. And there are also instances of bottlenecking where certain inbreds became popular during periods of time, leading to large proportions of the ancestry of that company emanating from a few key inbreds over time. And so what we're left with when it came to characterizing germplasm at Corteva was some shared ancestral haplotypes that go back to local linkage disequilibrium in these ancestral populations, but different family structures in terms of how that those ancestral haplotypes were organized. And then the other thing we're presented with was that these were genotyped on completely different marker platforms. And so, you know, even though there's common genotypes, common haplotypes there, you couldn't really see them because they were completely different markers analyzing them. And at the same time, the breeders immediately want to take advantage of everything that they're used to, the haplotype framework, genomic prediction, and so on. But the other ingredient that we had in our favor was we had some limited whole genome sequencing SNPs on both of them. And so this essentially allowed us to peer into a common SNP set that would allow us to determine how the different marker platforms characterize the common set of SNPs. And so essentially, I sort of took inspiration from the concept of this babblefish, if you've read The Hitchhiker's Guide to the Galaxy. But the idea of the babblefish is it can take in any sort of language, translate it to its common neural signals, and then output whatever the user, whatever the person needs to hear in their native language. And so by the same token, the idea is that we could use machine learning to take markers from any set of platforms. And because we had a set of common whole genome sequencing based markers across the different types of genetics, we could then translate it to this universal latent representation, which would then allow us to translate it into whatever we needed, whether we wanted to do marker imputation. So we would now have imputed markers on all the legacy platforms, whether we wanted to do haplotype imputation to see those nice colored diagrams across the diversity, or whether we wanted to do other sorts of applications like genomic prediction, population genetics, or even molecular functional inference. So what I'm showing here on the left is actually a visualization of this latent space where I've colored the genetics from one legacy organization orange and from the other legacy organization blue. The interesting thing here to point out was that you can actually see that for the most part, the genetics are fairly separate from one another, but you can see a few points where you get sort of an orange dot in the midst of blue. And this is actually a feature due to the way that plant IP protection goes. After a plant goes off patent, another company is free to sequence. And so what these are inbred lines from the legacy population B, the blue one, that were actually genotyped on population A's platform. And so what this is showing is that regardless of the marker platform that was used to genotyping, it's producing the same latent features. And so this is also showing this on the right. So what I'm showing in blue is the Euclidean distance, so just the squared difference between a particular line and itself under different marker platforms compared to when it's not the same. And this is the same except looking at the Pearson correlation. So in both cases, what you're seeing is that regardless of what markers were used to characterize the germplasm, it's producing the same latent representation that can then be translated into whatever you need it to be translated to, including markers. So ultimately what we did was impute both sets of legacy companies' markers onto all the lines so we could take advantage of all the previously existing germplasm characterization features and genomic prediction frameworks. And so ultimately what we saw is that across both legacy platforms, we were able to get marker imputation accuracy in a held out set above 99% accuracy, which let us have all the same resources across all the legacy germplasm, tens to hundreds of thousands of lines within a matter of a month or so. We could also use this to extend our haplotype framework to everything. So what I'm showing here are imputed haplotypes, so machine learning predicted haplotypes based on this common latent space. And you can also use those latent features directly for inference. For instance, this is the prediction of co-ancestry versus the true co-ancestry between genetic lines, but predicted directly from the latent space. On the right here is actually an example in wheat, where we predicted hybrid wheat yield directly based on the latent space instead of how it would normally be in terms of markers. The last thing I'd like to touch on with regard to how you can take advantage of a haplotypic framework is actually to augment your statistical power when it comes to making inference about molecular function. So because we have a closed breeding program, there's actually a lot of replication between our inbred lines when it comes to the haplotype level. And so what you can actually do is take advantage of this statistically, at least if you are willing to make the assumption that any molecular functional variation is due to essentially regulatory elements that are in cis. So for instance, on the left here, we have a replication of 2 for the red haplotype, 2 for the blue haplotype, and 1 for the green. Whereas on the right here, we have 1 for the yellow, 3 reps of the blue haplotype, and 1 for the green. And so because there's differential sharing of haplotypes between these lines, what it allows you to do is essentially automatically adjust for the influence of each sample based on the statistical power that imparts and the noise properties of that sample. And this came in use heavily when we were presented with some kind of weak data when we first got ATAC-seq for chromatin accessibility inference. But essentially what we were able to do through this haplotype pooling method is go from characterization of about 350 kilobases of total open chromatin sequence when we didn't take advantage of haplotype pooling to 5.2 megabases when we did include haplotype pooling. And we still see a highly significant increase in localization around known open chromatin peaks from an independent assay. And this included our, everything you're seeing in gray here is actually showing an open chromatin peak that was only assessed through pooling. So using this pooling technique, we were actually able to find alternative transcriptional start sites, and we also needed this open chromatin pooling to characterize a known QTL that's 65 kb away from our nearest gene. So I hope that I've given you a nice tour of how we're organizing genetic diversity at Corteva and shown you some of what we can do with the haplotype framework, especially when you combine that with modern machine learning methods. And that finishes my presentation. I'd just like to thank all my colleagues at Corteva.
Wonderful. Thank you, Eli. Are you able to see the questions that have been asked?
Let's see.
So I can actually read them to you so that we can make sure that our attendees can also hear them.
Okay.
Our first question comes from Mark Johnson, who wanted to know whether your process was locked into the genetic variation existing in your progenitor strains, or do you have ways for increasing and generating diversity?
So yeah, I guess there's two parts. So at the moment in corn, we essentially have a closed breeding system. So essentially the genetic diversity, at least the natural genetic diversity, would be coming from recombinations between existing germplasm. Although we do have diversity breeding programs where, say, we might bring tropical germplasm to North America. The other part of that, this might not be exactly what you were getting to, is using genomic editing technology to induce diversity that wasn't there before, or say moving something like disease resistance loci from tropical to North American germplasm. So that would be a way to sort of directly manipulate diversity. But for the most part, at least in corn, we're a closed breeding system. I see that there's a second question from Mark asking what the phenotypes that we're measuring. So, the principal one is yields, at least in corn. So that would be in terms of like bushels, how much grain you produce per acre. But we also want to have, say, low moisture corn, so you don't have to dry it down a lot. And a big thing that we spend a lot of time in at Corteva is making sure that the corn doesn't lodge and it doesn't fall down during storms. And so, we want our corn to yield a lot and, be resilient to the weather. So, altogether there's, multiple tens of phenotypes that we're measuring, but, yield is always the predominant one.
Fabulous. Well, I think that wraps up our time. Thank you so much, Eli, for your presentation.
Thank you.
And we're going to switch gears. Our third presentation will be given by Dr. Celia Payen from DuPont, and her talk is entitled High Yield and Robust Saccharomyces cerevisiae Strains for Biofuel Industry. So welcome, Celia. Thank you for being here, and the floor is yours.
Celia, I believe you're sharing the wrong screen on your computer.
Okay, let me try again. Sorry. We are not allowed to use Zoom. Okay.
Much better.
And then you should be able, okay, and I can start the video too. Okay, perfect. Well, thank you so much for joining. And it's perfect transition. We go from corn to ethanol. So that's very perfect. So what I'm going to tell you today is a very simple story on how we leverage tools that were built by the yeast community and how we use these tools in industry to make better product. So I'm going to talk about yeast, and my background is in genetics and genomics. So it is the only formula that I can understand. If you feed glucose to yeast, it will produce alcohol. That's great. We all love alcohol. There is a lot of applications for wine, beer making, and spirits. But today I'm going to talk about biofuels. And I'm going to talk mostly about the US market because that's one of the biggest. There is 200 plants, ethanol plants in the US, most of them in the Midwest. And they produce 16 billion gallons of ethanol per year. That's a huge amount. And it's also a very big market, $22 billion. What's great about this industry is that they love GMOs. GMO corn from Corteva, in the past DuPont, but also like engineered enzyme, and they are slowly switching from non-engineered yeast to GMO yeast. So on the graph below, you can see a simplified version of what an ethanol plant and the production of ethanol look like. We start with corn, We grind the corn, we cook the corn, and we add a lot of enzyme to extract all the starch and to convert this starch into glucose. And then the glucose is being used by the yeast to produce ethanol. Ethanol is going through the distillation columns, and that's what the plants sell. And that's what you put in your car when you fill your tank. There is 10 to 15% ethanol in the gas in the US, and up to 85% in other countries. And the leftover of the biomass from corn and yeast is being sold to the feed industry for cattle, poultry, and swine most of the time. Something that we cannot see really on this small graph is the scale of the fermenters. So if you look at this fermentation tank here where I drew a small yeast, most of them are 800,000 gallons. It's huge. It's the biggest fermenter that your yeast is going to be in. And that also leads to a lot of stresses that the yeast encounters during fermentations. There is osmosis stress, ethanol stress. Most of the plants go up to like 13–15% ethanol at the end. Some of them can go up to 20%. The media composition changes during the year because of the quality of the corn is different, but also between plants. So we need a product that can handle all of these changes. Some plants go very low pH because it's not sterile, so you need to slow down the growth of bacteria. And also, yeast produce a lot of byproduct during fermentation that can accumulate and be toxic. And then, as you can imagine, you cannot have 800,000 gallon fermenters inside a building with AC. So the tanks are outside. And summer in the Midwest are very hot, so you have a huge fluctuation and spike in temperatures. But also, fermentation is exothermic. And so there is also production of heat during fermentation. So yeast can handle most of these stresses individually, but here we are asking yeast to handle a lot of stresses together while still producing ethanol and finishing all the glucose. And here I want to show you real data from one of our customers. And we are comparing here data between winter in green and between summer in red. And you can see on the graph, and we plotted the ethanol being produced over time during one fermentation, so it goes up to 60 hours. You can see that during, if we compare summer with winter, you see a slight increase rate at the beginning, and then the fermentation stops fermenting. If you look at the bottom graph, we are not looking at the end of fermentation and looking at the glucose left at the end. The first few tanks, you see there is no glucose left, but there was few weeks where it was very, very hot. And you can see that there is 10 grams per liter of glucose left at the end. And all of that, just by increasing the temperature, you can see fermentation rate being different, lower ethanol yield, higher residual sugars, and that means that the plant is losing a lot of money. There is many ways of mitigating that. The plant can buy some capital equipment and run them, and we call them cooling watt towers. But just running these cooling towers yield to a cost of $500,000 a year just in electricity. And there's other ways to mitigate that. We can also deliver a more robust and thermotolerant yeast, and that's what we are working on. There's many ways of finding a better product, right? We could look at the diversity of yeast. There is some yeast that are naturally more thermotolerant, and we also look at mutations that make them more thermotolerant. And then by combining them, we can make robust products. So I show you the graph of what a plant looks like. But we still need to mimic the process in our lab. And to do that, we use different strategies. The first one is this yellow jug that I'm showing you in a picture. So we get the corn mash directly from our customers. And it's yellowy and kind of smell good, but also chunky and not clear. So we cannot really look at biomass and OD in this media. The second thing I show you in my first few slides is that when yeast produce ethanol, you also produce CO2. And so by using this NCOM module, we can track fermentation by just looking at the CO2 being released over time. And we also implemented fermenters, lab scale fermenter, to really mimic the whole process in our lab. So let's talk about robustness now. I show you that there is many stresses encountered by the yeast at the plant. And sometimes when we do screening, we find some yeast or mutation that increase tolerance to temperature spike. But then when we test organic acid, we see that the yeast is less tolerant. And that's not okay. That's really not what we want. What we want is like yeast seed that is more tolerant to all of the stresses. And that really will make a product more robust. Experiment. One of the first experiments I did when I joined DuPont 5 years ago after my postdoc, I leveraged this amazing tool that was developed by the yeast community a very long time ago, in which all of the genes were either deleted or overexpressed. And so the yeast community created this awesome collection of mutants. So I took these mutants, pulled them, and applied selection. In this case, I use the corn mash from the plant, and I added different stresses. And so the strain that have an increased fitness in this condition will take over the populations. And you can find them by sequencing the barcode, because each of these mutations was barcoded with a unique barcode. And that's an experiment that a lot of labs around the lab have done. And the barcode sequencing was developed in 2009. And this is the technology we used. But in this case, we needed to transfer the mutation from the lab strain into our strain. And so we used CRISPR to do that. And then we had many questions. We didn't know if, because different genetic background will have the same phenotype, but we also didn't know if the robustness will lead to more ethanol or just more biomass. So we had a lot of questions at the time. So here's just a simple graph to show you that most of the mutation, and that's been very well known for a long time, most of the deletion in yeast, in cerevisiae, are neutral. A little bit of them are deleterious, and very, very few of them are beneficial. And that's the one we are going to go after. Another way to look at the data is to look at this array, where on the y-axis, we look at all of the mutant strain. And then on the x-axis, you see the different stresses. And in this case, green is good and red is bad. And I'm sorry for the color. The marketing team asked me to use red and green in this case. And if we zoom in, you can see that some mutants are associated with increased fitness in two conditions or one condition, but not the others. But what we wanted is a mutant that increased the fitness in all of the conditions, like this gene that we found, the deletion of the LS1. And I will come back into this gene in a few minutes. So the first things we did, we took this gene, we deleted it using CRISPR in our production strain, and then we tested this strain in a different condition. Here I'm going to show you different temperatures. The graph on the left is in Fahrenheit because our customers use Fahrenheit, but the yellow bar at the bottom is 32 degrees, orange is 34 degrees, the top one is 37 degrees. And you can see this ramp. And this ramp is very important because this ramp mimics the average spike in temperature that is being seen at the plant during summer. And so in this case, we really mimic what's happening at our customer plant. If you look at the graph on the right, here I'm just showing the percentage of ethanol increase of the mutant over the wild type at the end of fermentation in the corn mash. And you can see that at 32 degrees in yellow, we see a 2% increase in ethanol titers, and etc. In all of the conditions, we see an increase. So this really showed that the deletion of this gene in our production strain can increase robustness at high temperature, but it also increase the titer at the end of fermentations. And in average, 1% means $1 million benefits for the plant. So it's a lot of money for them. We also tested this strain in different conditions and stress, and found that this mutation confer tolerance to high ethanol exposure and also dry solid, which means it's the composition of the corn mash. So by looking a bit more at the literature on what this gene was doing, so the DLS1 protein is part of the chromatin accessibility complex, and it has a paralog. So we are sweet, maybe we could find another gene. And so we deleted DPB3, and we found the reverse. You can see on the blue bar, we saw a decrease in production of ethanol at the end of fermentations. And so we found another paper in rice. So DPB3 exists in rice and all of the other species. And so in this paper, they found that by overexpressing DPB3, they see an increase in thermotolerance in rice. And so we decided to test that. But doing a deletion is easy. You just need to remove 1 or 2 copies. Overexpression is more complicated because you need to test different levels of expression. And in this case, we tested 5 different promoters. First, we added 1 copy. Then we use a promoter that increased by 12-fold over the normal promoters of DPB3. Then 28, 75, and 135. And we found that just increasing by 1 copy already increased the titer at the end by 1%, but the max that we got was with the 75-fold promoters. And so we took that strain and tested it in the different temperatures. And we were able to mimic the deletion, the phenotype found with the deletion of the LS1. And so that was quite surprising, at least for me, was that the deletion of 1 paralog has the same phenotype as the overexpression of another. And so the hypothesis is that when you delete DLS1, you disrupt the Shrike complex. And we know that the Shrike complex is important for the silencing of the telomere regions. And we know that there is a lot of stress response genes in these regions. While the overexpression of DPB3 captured DPB4 because it has a physical interaction with DPB4. And so there is less DPB4 available for the Shrike complex. And so it's also inhibiting the function of this complex. In this slide, I'm very simply summarizing subfunctionalization on bicomplex paralog, where you start from 1 gene, 1 protein that belong in 2 different complexes. Then you have the whole genome duplication that happened in yeast 100 million years ago. Now you have 2 genes, and then you have accumulation of mutations and separation of function between the 2 different genes and between the 2 different proteins. So this is just to show you how we use a tool that everyone knows very well and use within the yeast community to identify mutations that are beneficial in very specific applications. So in addition to that, we also have a team that work on pathway, and they're all pathway engineers. And they were able to design this beautiful pathway that increased the flux for the production of ethanol. And this pathway uses several genes from bacterial origins. And I cannot really talk about that. I'm really not a pathway engineer. But what we did is that we combined the pathway with the DLS1 mutations, and we produce a new product called Synertia Stripe GX. And this yeast also expresses a glucoamylase. And now I'm going to show you the impact of these beneficial mutation that we found on our product that we sell now at more than 70 plants across the US. And here I'm going to show one of the first. The plant trial started in June. The first 2 weeks were beautiful. It was nice weather, not too hot. And then it was horrible. It got up to 97 degrees in the fermenters. And the average time above 95 degrees was more than 12 hours. So very, very stressful for the yeast. But if you look at the graph on the right, you see that the glucose consumption during fermentations is very similar between early June and later June when it was hot, which means that our strain was able to finish all of the glucose at the end of fermentation, and that the rate of consumption was exactly the same as the same yeast, but during more normal conditions. And in this slide, I'm showing you the rate of production of ethanol over time. And you see again, the 2 graphs overlap perfectly. And when you look at the drop ethanol titer, so at the end of fermentation, how much ethanol was being produced, there is no differences between the nice condition weather and the very, very hot weather. And so that really showed that this product, Synertia Stripe GX, is very tolerant to heat stress. And so I show you a very simple experiment, very simple stories, but I hope you really illustrate that there is a lot of different needs, and that everything that you learn in academia and in the lab today during your PhD and your postdoc are very, very useful. And I was very surprised that I could apply that as soon as I joined DuPont. And from, chemical engineers and pathway engineers, strain construction and selection, screening, yeast production, we really need a lot of skills, and that's really what makes us strong. So I wanted to thank all of you, and if you have any question, I would be happy to take them now, or you can contact me later. Thank you.
Excellent. Thank you so much, Celia. We do have a few questions in the Q&A chat box. I'm happy to read them aloud so that our attendees are also able to hear them, or you can read them if you'd like to. It's up to you.
Oh,
Are you
yeah.
able to see the Q&A? Okay, why don't I give, I'll read them aloud and then you can answer them, okay? The first question comes from Mark Johnson, who says, you talked about improving the stress resistance of your strain. Do you also have ways to increase ethanol yield by reducing the amount of substrate in glucose that goes into biomass?
Yeah, so something that we do, because we have the fermenters, we can look at mass balance and look at carbon balance as well. So we know exactly where the carbon goes, if it goes through the ethanol pathway or to the biomass. And we know that the pathway increased the carbon utilization through the ethanol pathway, while some of the mutation we found impact the biomass. So we play on both. Yeah.
Okay. We have a question from Anya Oladart, who asks, do you have to screen many temperature-resistant mutants before you found one that didn't affect other important traits? And she said that you did a great job.
Yeah, thank you, Anya. I know her, that's why. I was very lucky. Really, it was my first experiment, and you know exactly what we do in Metria's lab. We picked like 10 winners. We make like 10 mutants, and the LS1 was the best, and we focus on that, and then we focus on the other later. So I think we got lucky there. But I think that testing in a lot of different conditions right away also helped do that.
Okay. A question from Narita Matra. How do you control copies by different promoters from overexpression of DPB3?
Yeah, so we do, there's two ways you can do that. You can do promoter swap using CRISPR. And so we just integrate a new promoter in front of DPB3. Or we just add another copy somewhere else that we know it's stable. So it's very stable, very easy. The yeast that we use is not an haploid, or it's not a baking or growing yeast. So it's easy and stable.
There is a question from your fellow panelist, Dr. Bruno. He wants to know, he says, robustness and heat tolerance seem important. Do producers repitch the yeast or start with new seed strain? Is it different in the summer?
Yeah, so no, they don't repitch. They always start the propagation tank with new fresh ADY or cream. Yeah, that's not like in the brewing industry where they repitch. There is some plant that do continuous process, and so that's a little bit different there. But for most of the plant, the ICM design plant, they just do batch fermentation.
Nice. Okay, we're gonna stop there. The questions will continue to live in the Q&A box. So Dr. Payen can answer them at a later date, but right now we're going to thank Celia for her presentation. And our next presenter is Dr. Julia Alterman from Atalanta Therapeutics, and she's going to give us her talk entitled Divalent siRNA Scaffolds for Robust Gene Modulation in the Central Nervous System.
There we go. Can everyone see that? Hopefully.
Yes.
Excellent. Thank you so much for inviting me. I'm really excited to be here and to talk to you about this project. Actually, this project hits close to home. It was a PhD project done in collaboration with a couple of postdocs at UMass Medical School, and that turned into a company. So I'm going to sort of take you through this story and tell you about how that happened. So as I mentioned, this all began at UMass Medical School with a collaboration between two professors, Dr. Anastasia Karova at the RNA Therapeutics Institute and Dr. Neela Ronan, who works on Huntington's disease. And when Anastasia first came to UMass, and this was maybe about 7 or 8 years ago, she was interested in making chemically modified siRNAs for the treatment of genetically defined disorders. And she went to Neela Ronan's office and she asked him if he had a disease that she could work on. And he said, well, how about Huntington's disease? So what is Huntington's disease? Huntington's disease is an autosomal dominant neurodegenerative disorder that's caused by an expansion of the CAG repeats in exon 1 of the Huntington gene. This mutation occurs in about 1 in every 10,000 people in the United States. And it can be seen as cell loss and atrophy in the striatum as well as the cortex. And so if you can see my pointer over here, this is a brain that's not affected, and this is a brain that's affected by Huntington. And as you can see, you have significant brain loss in both the striatum, which is here, as well as the periphery, which is the cortex. And symptomatically in patients, this looks like motor, cognitive, and emotional symptoms, as well as chorea, tremors, involuntary and abrupt movements, epileptic seizures, and depression. But it's actually a single gene that's causing this disease, a mutation in a single gene. And so this suggests RNAi therapeutics as a way to treat this disease. So what are RNAi therapeutics, and how might you use RNA interference to treat a genetic disorder? I don't think that I need to remind this crowd that the Huntington gene is going to make the Huntington mRNA, which is going to make the Huntington protein. But if we deliver something called a small interfering RNA, which is a small piece of double-stranded RNA into the cell, this double-stranded RNA can get loaded into a protein that's naturally present within your cell. And this protein is called AGO2, or Argonaut2. And once it's loaded, the sense strand, or the the sense strand will be discarded. This is also called the passenger strand. And the guide strand will be retained. And this guide strand has complementarity to the mRNA that you're targeting. So in this case, the Huntington mRNA. And once the guide strand is loaded in AGO2, this is called RISC, or the RNA-induced silencing complex. Once RISC is associated, it can go on and find its target mRNA, cleave its target mRNA, and this will lead to the target mRNA being degraded, and therefore the mutant protein will never be expressed. And then the RISC complex can be recycled and can go back and cleave additional mRNAs. But you hit a wall almost immediately, because there are many hurdles to overcome when using RNA as a therapeutic. And this includes the fact that it's readily degradable, it can cause an immune response, and it can be difficult to deliver to cells without packaging or some kind of lipid formulation. Fortunately, the RNA chemistry community has been working for decades on different kinds of chemical modifications that we can use to overcome these hurdles. And these include modifications to the sugars of the ribose, so 2' ribose modifications that you can see here, 2' fluoro and 2' O-methyl. And these not only prevent immune response, but they also stabilize against exo and endonuclease attack. We also use backbone modifications. In this case, I'm showing you a phosphorothioate modification, where one of the 2 non-bridging oxygens in the phosphodiester backbone is replaced with a sulfur. And these not only protect against exonuclease attack, but they also make these internucleotide linkages a little bit sticky. And so they will bind to proteins, and this can assist in allowing for protein binding on the surface of cells, and therefore these compounds can then get internalized into cells. Now this doesn't work for siRNAs because they don't have enough phosphorothioates in this region, but this is the mechanism that's used by antisense oligonucleotides for their delivery into cells. And lastly, siRNAs often use conjugates, and these conjugates can be different things such as vitamins or steroids, sterols. In this case, I'm showing you a cholesterol conjugate, which will again add something sticky to this compound, which will help with association to the cell membrane and internalization. And so as you can see here, we have primary cortical neurons that have been treated with siRNA passively, so not in any kind of lipid formulation. And you can see the siRNA here labeled with Cy3 labeled fluorescent marker. You can see that after 60 minutes, you actually get association of the Cy3 labeled siRNA with all of the neurites in your field of view. And when we look at silencing, we can see that the modified siRNA, and only the modified siRNA, shows potent silencing, whereas the unmodified siRNA is completely inactive when delivered passively. Okay, unfortunately, while these work really well in vitro, these kinds of hydrophobic conjugates do not translate very well in vivo in the brain. So what happens is when you deliver something that's very sticky or very hydrophobic into the brain, you'll have a lot of that compound retained at the site of injection, and then you'll have a steep gradient of diffusion away from that site of injection, which makes it hard for you to reach more distal areas of the brain. On the other hand, if you remove that hydrophobic conjugate, the phosphorothioates that are there alone are not enough to give you a significant amount of retention. But in fact, you do get a wider range of distribution as that compound is not hydrophobic, will not stick to the site of injection, and has a chance to spread out a little bit after the injection. So we came up with this idea of creating a molecule we call divalent siRNA, or di-siRNA. This compound actually has 2 siRNAs attached to one another with a linker. And what this does is it gives you 2 fully phosphorothioated tails. And these tails, both with their weak protein interactions, can act cooperatively to give you both retention as well as distribution of the siRNA. And so you can see in this picture that not only do we get significant retention, as you saw with the cholesterol, but we get widespread distribution away from the site of injection that encompasses the cortex on the side where the injection was. So this looks really promising. Next, we decided to see what kind of delivery we could get to the mouse brain after a cerebral spinal fluid injection. And so what we decided to do was inject into one of the lateral ventricles. The ventricles in your brain, you have a number of ventricles that are connected to one another that have something in it called cerebral spinal fluid, CSF. And this CSF moves through those ventricles and then will bathe the entire brain, go down the spinal cord, come back up. And so this means that whatever is in this fluid has access to lots of regions of the brain and spinal cord. So following injection of the di-siRNA into the lateral ventricle, which would appear right here, we first noticed on the right here, you can see that just when taking the brain out of the skull, you can see widespread distribution to both hemispheres of the brain that can be visualized in pink on the right here. And when you look at a sagittal section of the brain, you can see that we have distribution to the prefrontal cortex, medial cortex, posterior cortex, to the striatum, which is down here, to the hippocampus, to the thalamus, and even all the way back to the cerebellum. And so this distribution, while very promising, we needed to see whether or not this actually translated to silencing in these animals, and if this silencing had any kind of duration of effect, because we expect that with increased modification, we could potentially increase the stability of our compounds and therefore the duration of effect of these compounds. And so the experiment we designed was to inject 475 micrograms of compound, ICV, into the lateral ventricle. And then we injected both a non-targeting control. So this is an siRNA with the same chemical scaffold, but that does not target any gene in the genome. And we also injected a Huntington targeting siRNA. And we took the animals down at 1 month, 4 months, and 6 months post-injection. And we looked at silencing in the medial cortex, posterior cortex, striatum, hippocampus, and thalamus. And what we found was very exciting. We can see here that after 1 month, we have undetectable levels of Huntington protein silencing, suggesting we got almost complete Huntington protein silencing in all of the regions that we looked at. And so this includes, as I mentioned, hippocampus, thalamus, striatum, posterior cortex, and medial cortex. And you can see that even as you go out to 4 months and 6 months, we're still maintaining significant silencing in many regions of the brain, although in the posterior cortex and the medial cortex at 6 months, that efficacy becomes much more variable. But this was very promising for the use of this compound, if you can think, for very intermittent dosing. So it's great to see that this compound worked really well in the mouse brain. But unfortunately, as you know, the mouse brain is quite a bit smaller than the human brain. And so it's really important to determine whether or not what you saw in a small animal brain can actually be used to get you widespread distribution in a much larger brain. So in order to test this theory, this hypothesis, we decided to use non-human primate brain, specifically the cynomolgus macaque. So the design of this experiment was as follows. We injected unilaterally ICV, 25 milligrams of the compound, and we looked at compound delivery, efficacy, gross brain images, inflammatory response, systemic toxicity, and off-target effects. So as you can see here on the upper left, that after a single unilateral injection, the entire non-human primate brain is shown pink here with the compound. And one interesting thing that this tells you is that a unilateral injection of this compound into a larger animal brain allowed for bilateral distribution of that compound, where you can't even actually tell which side was injected if you couldn't see where the injection spot was. If you go into slices of the brain, you can see also that the entirety of the cortex is stained with the compound, and it's also penetrating into deeper regions of the brain in the striatum as well as into the hippocampus. And if we zoom in a little bit into some of these regions, we can see that all of the cells in the field of view, either in the cortex, the caudate, or the hippocampus, seem to be associated with this compound and have internalized this compound. We also wanted to see what kinds of cell types were associated with the siRNA. And what we found was when looking at markers for neurons, so this is NU-N, or markers for glial cells, which is another cell type in your brain, this is GFAP in purple, that you can see that the compound associates both with neuronal cells and with glial cells. And so what this means is that if you have a genetic neurological disorder that's affecting different cell types, you can in fact use this scaffold to treat and silence that gene of interest in different types of cells. And lastly, but most importantly, all of this did translate into silencing in the non-human primate brain 1 month after injection. And so what you're looking at here is 4 different regions of the brain, the 4 regions that we tested. So this is the cortex, the hippocampus, and the caudate and putamen, which make up the striatum. And we tested both the ipsilateral side, so this is the injected side, as well as the contralateral side, which is the non-injected side. And as you can see here, we have significant silencing in all of the regions of the brain that we tested, and that we can, in fact, get pretty comparable silencing, both in the injected side as well as the non-injected side. Next, we move to the spinal cord. And similar to the brain, you can see that all the regions of the spinal cord have retained the compound. So this includes the cervical region, which is at the top here near your neck, the thoracic region, and the lumbar region. And if we go in further and look at NU-N or neuron cells or GFAP, you can see that again, these large motor neurons here and the glial cells have retained the compound. And of course, this translates to silencing both in the white matter and gray matter of the spinal cord in all regions of the spinal cord, the cervical, the thoracic, and the lumbar. I also want to show you just an overview that there was no overt toxicity in non-human primates, although I don't have time to go through all of this data. But the kinds of experiments we did where we looked at gross brain changes. So this is MRIs pre and post, so preoperatively and then 30 days after the injection. And you can see that there are no gross changes in the animal brains. We also looked at inflammatory responses. So there's a couple of markers for inflammation in the brain, and none of those were significantly increased. We looked at systemic toxicity. So these compounds are going to be cleared through the liver and kidney, and we wanted to make sure that we hadn't done any damage to those organs. And as you can see here, there was no significant increase in these. And lastly, we looked at off-target effects, so RNA-seq. So one of the things that I want to point out about this siRNA scaffold, or really about informational drugs in general, and so oligonucleotide drugs we consider informational drugs. When you think about a small molecule's chemical structure contributes both to its delivery and distribution, as well as to its target specificity. So you can't adjust one piece of that chemical structure to increase delivery and distribution without affecting its specificity to its particular target. However, in the case of informational drugs, it's that chemistry I originally told you about, the backbone chemistry, the conjugate, the internucleotide chemistry, that's really driving the delivery and distribution and PK properties. Well, it's the sequence that gives you target specificity. So what this means is once you've developed a chemistry or a chemical scaffold that delivers beautifully throughout the brain, you can then put in new sequences in order to reapply that scaffold to another genetic disease that you're interested in targeting. And so that's what I'm showing you here. This is a different target. I'm showing you also significant silencing after 1 month in the hippocampus, thalamus, striatum, and posterior cortex. And with that, I'd like to summarize. So as I've shown you, siRNA shows widespread distribution and long-term efficacy in the mouse brain. siRNA distributes and silences throughout the non-human primate brain and shows a positive safety profile. siRNA can be adapted to target any genetically defined neurodegenerative disorder in theory. And most importantly, academic research holds great therapeutic potential. So no one forget that. There are lots of people to thank for this project. It was a collaboration between a lot of labs at UMass, specifically Anastasia Karova's lab, Marian DeFilia's lab, Alpha Pai's lab, Neil Aronen's lab, the whole monkey team. And I'd also like to thank, of course, Atalanta, the company that was formed out of this project, and Atalanta's founders, who were Anastasia Karova, Neil Aronen, and of course, I'm sure all of you know Craig Mello. With that, I would be happy to take any of your questions.
Fabulous. Thank you so much, Julia. That was wonderful. We actually have several questions for you. The first comes from Kashava Mysore. I think that's how you say it. She wants to know what is the length of the siRNA and does it matter how long it is?
Yeah, so the particular siRNA you're looking at here has a 20 nucleotide guide strand and a 15 nucleotide passenger strand. Typical siRNAs are about 21 nucleotides in length. Turns out that you do have some flexibility, a little bit of flexibility by 1 nucleotide or 2, but generally what fits into AGO2, since this is protein-assisted silencing, is around that length. So most of them are right within that short length range.
And then a follow-up question to that is there a relationship between length and off-target effects?
Not really, and I'll tell you why. Because off-target effects really are based on the region, the sort of seed region, or the region that is binding to the mRNA. And it's been shown that really it's only nucleotides 2 through 17 in that oligonucleotide, if you look at the crystal structure, that can bind to any mRNA, whether it's your on-target or your off-target. And so extending on the far end, so on the 3' end of that guide strand, is not likely to give you more off-target effects.
Okay.
So I think that's
So that makes sense. An anonymous attendee would like to know whether you can imagine a delivery route that is less invasive than ICV.
Yeah, so what we're looking at, I mean, this current compound does not cross the blood-brain barrier, so it has to be delivered into the cerebral spinal fluid. The route of administration that we'd be interested in pursuing would be intrathecal. So there are actually a number of drugs that are delivered intrathecally, and you can imagine a drug like this you would only receive once every maybe 6 to 9 months.
Julia, can you explain what intrathecal delivery is?
Yeah, sorry. So any of you have heard of a spinal tap? So they go in and they inject it just in between 2 of your, I've actually had one. It's not painful and it's relatively quick. And so it's just injecting it directly into the CSF in your spine. And this is considered a very good route of administration for any kind of drug for the brain. An additional version of delivery that can be used is something called an Ommaya reservoir, which has been used for cancer and pain patients. And this is a reservoir that would be put into under the skin, between the skull and the skin, and then can be driven down into the lateral ventricle. And then I guess at home or just at the nurse's office, you could go and get injections directly into that reservoir. I'd remind people that the diseases that we would be treating, Huntington's disease, Alzheimer's disease, ALS, these kinds of diseases are horrible. And some of them cause rapid degeneration. And so many of these patients are willing to take any kind of route of administration that would allow them not to suffer through the diseases that they've watched their families suffer through.
Absolutely. Actually, to that end, I have a similar question. You described a lot of beautiful work in the brain. Is there any utility for this scaffold for systemic delivery for other applications that are not neuronally based?
There are potentially uses for this scaffold systemically, but at this point in time, Atalanta is focusing solely on neurological genetic disorders.
Great. I'm going to take one more question before we wrap it up. Toshi Banerjee wants to know whether or not you check the stability of the siRNA in the primate brain.
Yes, so we did do, we didn't do full-on stability studies looking at mass spec, but we are able to detect the compound in the brain after 1 month, so that we know that some of the full-length compound is still there. But we would need to do more long-term stability studies when we do preclinical studies.
Okay, great. Thank you so much. And like I said, the rest of the questions that were asked will be available to Julia so that she can answer them later. But right now, we're going to thank Julia for her presentation and have our last presenter, Dr. Janis Weeks, take the stage. She's going to give us a talk about her work at NemaMetrix entitled NemaMetrix, Cutting-Edge Genome Editing and Phenotyping Tools in C. elegans and zebrafish, and how we've targeted parasitic worms. So with that, the floor is yours.
Thank you. Can you see my screen? Yes, hello?
No, not yet.
Not yet. Okay, I know what I have to do. Hang on. Share screen. There we go. Okay, is it there?
Not yet. There should be a box that pops up after you hit share screen that lets you choose what screen or app to share.
Oh, okay. There's, oh, I see. Share screen, that screen. Share.
There we go.
That's looking much better. Just go into presenter mode. Yep, we can see you.
Perfect, thank you. Well, it's a pleasure to be here with all of you remotely. Let me start by saying, first of all, our company's name has changed. It was NemaMetrix, and now we're In Vivo Biosystems, and I'll say a little bit more in a bit about this. I also want to take this opportunity to just mention that the work I'm going to talk about was done with a lot of other people, and some of the PI's names are listed below, and I'll also mention them, try to mention them as I go along. Okay, I'm going to give a little history of our company. So 1988 and 1993, I and then my colleague Sean Lockery both came to the University of Oregon as faculty members. Whoops. And so Sean's lab works on C. elegans neurobiology, and I've been an insect neurobiologist most of my career. And then we're going to skip a lot of the details in between, but in 2011, Sean and I spun off a company, NemaMetrix LLC, to commercialize a new device for C. elegans electrophysiology. So in 2016, we converted to a corporation. 2017, we acquired Nudra Transgenics in Salt Lake, and that brought in a lot of just key expertise in C. elegans and zebrafish transgenics. Recently, we've had over $1.5 million in revenue, and we've brought in over 10 million in investor funding and NIH grants. And then just last month, we converted to a new name, In Vivo Biosystems, to reflect our broader focus. So NemaMetrix came from nematode, which is what we started with, but now we work on other species as well. And we have about 50 employees right now. So what do? We specialize in C. elegans and zebrafish, and we offer what's listed below here: custom transgenics, extensive phenotyping, deep data mining from the phenotype data, and we can couple all these with drug and compound treatments. And our customers include academic labs, pharma, nutraceutical, and biotech companies, and then we also work with clinicians. And one major strength of ours is to make what we call clinical avatars. So we insert disease genes and variants into either worms or fish, and we can create human models of human disease genes. And for any of you out there who are NemaMetrix customers, we just want to reassure you that we still have all the usual instruments, reagents, and services that you're used to from NemaMetrix. So now let me talk about the core technology with which we founded NemaMetrix, which was a higher throughput method for recording pharyngeal pumping in C. elegans. So let me say a little about the pharynx. Here's the C. elegans, here's the front end. The pharynx is highlighted in green. And up here you see, based on the colors shown, that there are various cells in the pharynx. There's muscle cells and neurons. And so the pumping of the pharynx is generated by coordinated activity in the pharyngeal neurons and muscles. And the function of the behavior is to ingest bacteria for feeding. So this behavior is really important for feeding, but it's also turned out to be just a brilliant readout for all sorts of aspects of worm physiology. So it's been used as a readout for health span, lifespan, a variety of human disease models. This data here, these data are from my lab on an Alzheimer's disease model, serotonin signaling, oxidative stress, toxicology, and all sorts of aspects of neurobiology. So pumping, you know, the utility of pumping goes way beyond just getting food in the worm's mouth. So how to count pump frequency? Traditionally, it's been done by ways that you see here on the left. So someone just watching a worm in its pharynx and counting the number of pumps. And these counts are typically done for, say, 30 to 90 seconds or so to find out a worm's pump rate. But in the mid-90s, Raisin and Avery developed this amazing technology to record electrically the pumping rhythm. And these recordings are called electropharyngiograms, or EPG, and they're completely akin to an electrocardiogram and how those are recorded in a human. So for an EPG, a worm's in a saline bath, its front end is sucked up into a pipette. There's an electrode here and an electrode in the bath, and then we record the voltage differentially from those two electrodes. And here's the kind of recording you see during pumping. And for each pump that the worm makes, it starts with an E spike and is terminated by an R spike, which represent the excitation and relaxation of pharyngeal muscle. And then some of these other deflections are specific synaptic events that help control the rhythm. So this is an excellent technology, which is still used, but it's a bit slow. So you record one worm at a time, and you have to chase the worm around the dish to get him in the pipette. So the question was whether one could increase the throughput for EPG recordings. And that brings us to this device, which was invented by Sean Lockery. It's now patented, and it's a microfluidic device. So it has a glass slide at the bottom. Then there's a layer of a silicone elastomer called PDMS. And in the PDMS, there are these little channels. We've put dye in this picture. So the worms are put in here in the input port, and then we gently propel them, one worm in each of 8 recording modules. And then after the worms are in, and they're in saline, of course, buffer. And then we put an electrode here in the input port as a reference electrode, and it also serves as the conduit for perfused substances that we apply. So we record from 8 worms while perfusing. We have custom software that extracts and analyzes the EPG data. And one advantage is we can record for hours. So you can record the same worm for hours and hours in baseline conditions, and then in the presence of the drug, and so on. This is a picture of a worm in a recording module. So there's a little narrowing of the channel. In this case, we're head first in the worm trap. We see the classic EPG recording. And with the 8-channel platform, we can record from 8 worms at once, as you see here in different colors. And you see pretty active pumping activity, so 4 to 5 hertz or pumps per second. And that's because in the experiments I'll show you, we stimulate sustained, robust pumping in the baseline condition by including in the perfusate either serotonin, 5-HT, or E. coli to stimulate pumping. All right, now I'm going to change gears. I was asked specifically to talk about some of our work with parasites, so I'm now going to switch gears to that. And this is just a picture of human hookworms shown in situ feeding on intestinal epithelium. All right, you may know, and I should also, I just wanted to say that my interest in parasitic nematodes and global health has been prompted by having spent over 20 years working in Africa, developing education and research capacity, excuse me, in neurophysiology and neuroscience. So I have a particular interest in this. So parasitic nematodes cause a staggering disease burden globally in humans, but also in animals, which affects human health. Infections in kids, and here's a little girl in Zimbabwe. Here's the kind of worm load that a child may carry. And not surprisingly, these worm infections can cause anemia, malnutrition. It impairs kids' physical and cognitive development, their education and productivity. So it's a huge global problem. There are drugs, they're called anthelmintic drugs, which have been effective in many ways, but they have shortcomings. And the big specter is ever increasing parasite resistance to the drugs. So we need to develop new anthelmintic drugs. And as a neurophysiologist, what's always interested me about this is that most of the anthelmintic drugs target ion channels and neurotransmitter receptors in the worms. So here's three of the main anthelmintic drugs. Ivermectin acts on glutamate-gated chloride channels, levamisole on acetylcholine receptors, and piperazine on GABA receptors. And these ion channels and transmitter receptors are still considered valuable drug targets. But a barrier to progress in this area was that the field lacked an easy way to test candidate drugs' ability to mess up electrical activity in the worms, to perturb electrical signaling. So that was some of the key motivation for Sean's invention of the 8-channel EPG chip, was to increase the throughput and help facilitate drug development in this field. And of course, the technology has gone on to be used in many other ways as well, but I'm going to focus today on anthelmintic drugs. And this animation just shows a typical recording. What we see is 7 worms here pumping away in control conditions with serotonin. Then we switch the perfusate to ivermectin, and you can see that pumping stops rapidly. So pharyngeal pumping provides a really robust, sensitive readout of anthelmintic bioactivity. All right, what have we done with this technique? So here's a study where we showed that this technology in EPG recordings could distinguish C. elegans strains that were either resistant or sensitive to different anthelmintics. We, on another project, screened a drug library of FDA-approved drugs to look for new anthelmintic candidates. Interestingly, and we used a number of assays, including EPG recordings, the top hits were all psychiatric drugs, so antidepressants and an antipsychotic. And then we went on beyond C. elegans to show that these same hits also killed three species of parasitic worms. Okay, so we've done various things with the EPG technology and C. elegans, but C. elegans is different in important ways from parasitic nematodes, and there's actually some controversy in the field about the extent to which C. elegans is a good model for looking for anthelmintic drugs. So my question was, could we modify the EPG platform for use with some of the worms that we actually want to kill? And fortunately, we were pleased to find the answer is yes. And this is work for these human soil transmitted helminths funded by the Bill and Melinda Gates Foundation. And then we also did work with Adrian Wolstenholme at Georgia, funded by the National Center for Veterinary Parasitology, on Homunculus contortus, which is a really serious parasitic worm of goats and sheep. And here you can see the profound anemia in the whiteness of the conjunctiva in this goat's eye. All right, we made this work. Here's a picture of a human hookworm host stage in a custom EPG chip. So here's the head. Notice how different the head looks from C. elegans, pharynx. This worm is tail first in the worm trap. And we were gratified to see, the standard pumping activity that we're familiar with from C. elegans. We went on, I'm going to stick with ivermectin is my representative anthelmintic drug in this talk today. So here's just an example of ivermectin inhibiting pumping in a hookworm. Looks pretty similar to the C. elegans data. The C7 worms are pumping away. We switch to ivermectin and pumping stops. And, you know, this may look kind of easy. It's like, well, why wouldn't it work? But in fact, there's some tricks going from C. elegans to working with these parasites. We had to fiddle with the chips, figure out what kind of culture medium the worms wanted to be in, what kind of additives would make them pump. Did they want serotonin? Did they want blood serum? We had to fiddle with temperature and then look at how we sourced the worms. Were they pulled out of a mammal or were they cultured in vitro, in culture. And then just another example, here's Ascaris another type of parasite. Again, we were able to elicit robust baseline activity in these worms and then show that the activity was inhibited by anthelmintics. So basically, we were able to validate this platform for use with actual parasites that we want to kill. We've also gone on, in this case, this is work done in collaboration with John Hodden's lab at George Washington. We've gone on to use these EPG recordings in hookworm to screen plant products as potential anthelmintic drugs. And in this case, we focused on Mamortica carrantia or bitter melon, which is used traditionally as a worming tea in many parts of the world, including, say, Haiti. And these are just EPG recordings in control worms. These are worms treated with aqueous lettuce extract, which was our, benign plant test product. And then finally with a similar extract from Mamortica. And you can see by 4 hours, the pumping has stopped. And this precedes by about 12 hours, the time when the worms actually die from ingesting this extract. So we've shown that the method is good for screening drug libraries, but also can be used to screen natural products. Final thoughts here. First of all, spinning off a company from a university is definitely an adventure, but doable and gratifying. And anybody out there thinking about doing such a thing, I'm happy to talk to you about what it was like. So Sean and I started NemaMetrix with this core technology of the EPG chip, but what the company does now has just grown incredibly, way beyond what we could ever imagined. And notably, we expanded our transgenics from C. elegans and zebrafish, so both the transgenics and the phenotyping. I spoke specifically about the work validating the EPG platform in parasitic nematodes. And this platform now is being used for anthelmintic drug research. And another way in vivo biosystems is involved in finding new drugs for parasitic disease is we can swap parasite genes into C. elegans. So you just take the parasitic gene homolog, ortholog, say an ion channel, and put it in, replace the C. elegans version of that gene. So the future, we've always relied heavily on input from the community about what we can do to make your research better. So anybody out there, get in touch with me. If there are instruments or reagents or transgenic animals or phenotyping and data mining services that would help your research, we would love to hear from you. And let me just finish by thanking you for your attention. And here's some photos of our leadership team.
Wonderful. Thank you so much, Dr. Weeks. We do have a question from Mark Johnson. He wants to know whether you can tell us a bit about what you're doing with zebrafish, perhaps what it is you're studying.
Yeah, I actually have some slides on that. So our zebrafish genome editing, let me go through here. We do site-directed mutagenesis of various types. Let me just put this up in presentation mode. Knockouts, point mutations, deletions, we can flox alleles. We can construct fish with flags or tags, fluorescent tags, epitope tags. We can knock in different expression lines. And then we also are set up to do phenotyping as people wish.
Great, thank you.
Mhm.
I actually have a question, not necessarily about the work itself, but you alluded to the idea about starting a company after being an academic scientist, and that was sort of the main focus or the goal of these sessions, was to highlight that transition. Do you find that there's one that you prefer, or that there are challenges or benefits to working in one atmosphere over the other? Are you happy that you made that transition into an industry setting?
Well, I never imagined I would go into industry. I mean, that was never really an intention of mine. But then as it transpired, I thought, what the heck? It seems like, something different and interesting, and we can contribute. But yeah, there's a lot of differences in academia and being in the business world. It's also hard. Sean has stayed a faculty member at U of O, and I retired 2 years ago, so I could focus more on the company. But yeah, so there are pluses and minuses really for both kind of position. But for anyone interested in like challenges and testing one's limits and learning new things, I found it just incredibly exciting.
And maybe I can open this question up to the rest of the panelists. You've all done a fantastic job today of describing your work in industry and explaining how the work that we all learn in grad school can be applied to real world applications and benefit not only, to further research on a fundamental level, but also to apply to consumers and to patients. And so if any of you have any thoughts on this, I would welcome your input. That is certainly why we're here, like I mentioned. If there aren't any other comments, or Celia, did you have anything you wanted to add?
No, it's very interesting, your question, because I did not realize that my skills that I was building during my PhD and my postdoc could be useful.
Yeah.
And so when I saw the application, when they were looking for someone specialized in functional genomics and evolution at DuPont, I was very surprised, and that made me excited to apply that position. And it's been useful. They really need a lot of different skills. And so I highly encourage everyone to apply for position and look at what's happening in industry.
Yeah, absolutely. I think that was sort of one of the things that we were hoping to get across, is that there are positions and opportunities in industry for such a wide range of sciences that I think you'd be surprised how much you can do with your education or your graduate school or postdoc experiences that would apply to a wide range of utilities and translations. And I think that being in an industry setting for so long, I myself have worked both at Genzyme and now at Atlanta for almost 20 years. You get so accustomed to the familiarity with that field and knowing how your work can be applied. But if you're an academic scientist by nature, it's not quite as obvious to you how to apply your experience and your education to a more translational approach. And so the reason that we wanted to undertake the sessions was specifically to highlight that those opportunities and to show work that was meaningful and interesting, but could still apply to both an academic and an industrial setting. So I'm pleased that even though we had to transition to an online format, we were able to get that message across and to bring these speakers to this format so that we could still highlight the work that's being done. And we were fortunate enough to have speakers from each focus and application to kind of talk about a wide range of uses. We had our clinical application and our process improvement, engineering, and even some that's still, although there is an industrial application, they still focus very much on the academic questions being asked so that the entire field can be furthered globally, not just with a focus on one particular customer or audience. And I think that that's really an important thing for students and for postdocs to be aware of. That especially with differences and changes in funding over the last 10 years, I know faculty positions have become more difficult to come by, and those opportunities to do exciting cutting edge basic science still exist in a range of settings, not just as a PI in academia. I open the floor up to the panelists for any last thoughts.
Looks like Mark was asking a question.
Yeah, Mark Johnson wants to ask the attendees how the GSA could help. Okay, so wondering how the GSA could help industry scientists and how they can help serve industry scientists. So if any of you have thoughts on that, please speak up.
I mean, I'll say something real quick. I think they already do quite a bit. I was in the national lab for 10 years before starting at Zymergen, so it's quasi academia. But there's always, in the fungal world, there's definitely industrial people that go to that, and it's a couple of companies have been around supporting that fungal conference for quite a while, like Novozymes, and so it's just definitely we're there. Sometimes companies don't present posters as often. I think maybe if we encourage and make it safer for them to get permission. I heard at least one other speaker say, my legal team wouldn't let me show this or whatever. There's something about that would be helpful. But yeah, I think the GSA is doing okay with it. It's doing pretty well.
Nice.
I would second that and say, as much as you can invite industry and invite people that work at companies that utilize these kinds of techniques in, so that they can walk around at poster sessions and introduce themselves to postdocs that are looking for jobs, so that they can job post. Maybe when they give a talk at the end and say, let us know if you're looking for a job in X, Y, or Z. Any way to introduce people in the basic sciences, students and postdocs and even professors who could be interested, to those people on the other side, because sometimes, they're just going to their separate conferences. I think it's really important. I know that in the RNA field, the RNA Society has opened up a session in collaboration with the Oligonucleotide Therapeutics Society. So typically the RNA Society meetings are very basic science based on like the science of RNA. So you'll learn about splicing, or you'll learn about whatever basic RNA functions, but when you go to the Oligonucleotide Therapeutics Society conference, you learn about how to use oligos as therapeutics. And the two were having two separate conferences. And now they have a swap where they each do a session from the other's conference and invite those same people, so that the people at each of those conferences can get introduced to basic science as well as to the people in industry. And so, I think reaching out to other genetics conferences that are maybe more industrial focused could be valuable.
I totally agree, and it also opens up the opportunity for collaboration, which Julia, you were able to show us beautifully, how something that starts in an academic lab, or even as a graduate school program or project or a postdoc project, can eventually translate into either a meaningful collaboration with translational clinical application, or in your case, a company. So thank you all very much. I just want to put up my last slide to thank everybody. To thank the speakers today, to thank our sponsors at this session, and to thank my amazing colleagues at the GSA, Mark Johnson, my co-chair, Anne Marie Mahoney, Susie Brown, and Erin Suderman for supporting these sessions and for helping us to get this to the online format, and for being so wonderful to work with the last couple of years. I very much appreciate your attention and for attending our session, and please reach out if you have any questions going forward, or if you'd like more information about what we do. Thank you so much, and take care.
Thank you.
Thank you.
Bye-bye.
Good night, everyone. Thank you.
Thank you.